So Parkinson's disease is a devastating neurodegenerative disorder that is mainly characterized by motor symptoms and is thought to be caused by a complex interplay between genetic risk factors and environmental risk factors. We really don't know the exact causes of Parkinson's disease, and although we have good symptomatic treatment, going forward in the future for disease-modifying therapy, we would have to understand really the molecular mechanism of disease. The study will be published in Brain, which is a leading neurology neuroscience journal in the field. And I think it will be impactful in a way because we identified a novel risk factor for Parkinson's disease that plays a role later in life. By now, we know about 20 genes that have been shown when mutated to cause Parkinson's disease, but those monogenic familial forms only account for maybe 5 to 10% of known Parkinson's cases. The majority of the sporadic Parkinson's cases, we just don't know the causes of those. From the known causes that we know today that really can cause Parkinson's disease, those genes can be basically categorized into two classes. Those are dominantly inherited forms or recessively inherited forms. It means we carry two copies of each gene in our cells, one from the father and one from the mother. And in the dominant cases, a single mutation in one of those copies usually causes disease. For the recessive cases, one would have to inherit two mutations in both of those copies in the gene to really cause the disease. What we found is that although we know that mutations, the gene we're talking about is called pink, although we know that in pink, the gene we're talking about is typically associated, two mutations are typically associated with Parkinson's disease, we found that a single mutation, just one of the copies of the pink gene, can lead to Parkinson's disease, although later in life. The implication for the patients will be yes, because so far we know these early onset patients that have two mutations in the gene get the disease maybe around with an age of 45 years or younger. But as said, for the later onset cases, we still don't know the mechanisms. So having identified this risk factor for Parkinson's later in life would give us the possibility to screen patients and to see if we can identify more of those risk factors to elucidate the causes of late onset Parkinson's disease. It will be critical to understanding really the molecular and cellular mechanisms of Parkinson's disease, the gene networks or protein networks that cooperate within the cell, and based on this understanding of the mechanisms, then to develop biomarkers and therapeutics. So pink and Parkin, that's the second Parkinson's genes, they basically cooperate in a pathway and identify damaged mitochondria within a cell as part of a quality control system. And mitochondria are small organelles in the cell which generate energy that is essential for almost any enzymatic reaction in the cell. So it's important to have a system that would identify, label, and target damaged mitochondria for degradation. If this neuroprotective pathway would fail, then damaged mitochondria would accumulate, become harmful to the cell, and eventually could cause cell death. So a detailed understanding of this protective pathway that is actually very complex would certainly help generate new rationale for drug design for the future for disease-modifying therapies.